information there. So I was planning to visit some relatives in Europe, and and I asked for some advice, from some of these students and colleagues. Museum, and because at that time I was very much on trilobites, he said you should ask Dr. Richard Forty, which I did. So a lot of time passed, and now we are in the 1990s. Pennsylvania trace fossils in the Mid Geological Survey, and I get these strange trace fossils. Like wingless insect trace fossil. So why don't you ask this guy at the Smithsonian for Conrad Labandeira? So here I am, I contact him, and out of this, working on these insects that we have met before, until I got some of the hard copies of the paper with tiny little notes and corrections and suggestions by the side of the paper. Writing, I have seen it before. And then I asked him, by any chance during the late 80s? And he said, yes, I was working on trilobite at that time. And so that's a funny story. We reconnect almost 18 years later, and we know that we have met really, really young. So since then, a very close collaboration, and I want you to tell you that he's not only an extraordinary person, very generous. He has over 100 graduates, postdocs, volunteers who work with him. So I don't take more of your time. I leave you with
Thank you, Gabriela. That was the point. Today is research that takes me all the way back to the Devonian, but it really runs the entire gamut of the fossil record. And so I will out to the students on the right that were more recently involved. And at the top is, she's now, this is Lifang Zhao, and she's doing at the Institute of Zoology in Guangzhou. David Paris from Spain, I've done a lot of work with, and we'll see some of our work, and he's in Barcelona, University of Barcelona. And the third student is Xiaodan Min, and we have worked on many fossil insects that were pollinators, and she's now at the University of Hainan, also in China. So what I'm going to discuss is the summary slide right here, and this is everything that I'm going to talk about is going to lead to this. This is what has happened during the ascendancy of angiosperms and how pollinators responded to that. I'll be talking at a more topic where we look at the herbivores being across this very phenomenal event during the early Cretaceous. So I'm not going to try this. No. Move it this way. Okay, thank you. So I'll talk about how do we use evidence for pollination. What is indirect evidence? What is direct evidence? And then we'll start in the Devonian, where maybe the early precursors to pollination were established. Not that these were pollinators, but that they were feeding on the reproductive structures of primitive vascular plants, particular spores, and that later on we run into the consumption of pollen. And then by the time we get to the Permian, much later in time, we actually have good evidence for pollination. And then during the mid-Mesozoic, everything breaks loose with these different modes of pollination. And then especially pollination mode 3, which is the long proboscis mouthpart syndrome or mode, as I prefer to call them, and with these 4 orders of insects. And then we'll talk about the plants and give 2 examples, one in which the plant is featured, and then another one in which the insect is featured, and about the intricacy of these pollination syndromes before the appearance of angiosperms. And then we'll talk about some transitions into angiosperms, and what are kind of the macroevolutionary patterns that we see in the fossil record, and that'll be summed up. So how do you tell evidence, indirect versus direct evidence for pollination? So there are 4 types of indirect evidence. You can look at morphological structures of the head of the insects. In this case, we're looking at long proboscis insects, or we can look at the plant part of this. We can look at structures such as nectaries, such as other attractants to plants that offer rewards to insects. And then we can also look at the coprolite record. So here is a coprolite from the Calhoun coal. It's anatomically preserved. You can actually see the individual pollen grains. It's a cordite. It's 100% cordite pollen, a thing called Florinites. We'll talk about that later. And then there's also the evidence of the damage that potential pollinators leave on flowers. This is from the Dakota Formation of Nebraska. It's a petal of one of the earliest flowers that we have, for which is good preservation. And you can see some of the damage along the margin of the petal, probably done by thrips. But there's also other kinds of damage that we'll see at the end of the talk on these plants. Oh, and the direct evidence is this example from Oliva Amber from the early Cretaceous of Spain, in which we have an insect that landed in a viscous resin dollop and actually went through as it kind of struggled for existence, leaving a wake of pollen, these little dots of pollen grains, and they're enlarged here. So in a sense, we're actually catching the insect in the act. Not of pollinating, but of actually in the process of pollinating and shedding all of its pollen grains as it traveled through this viscous mass of resin. Let's talk about the late Paleozoic. And this is from the Welsh borderland of the UK. And these are coprolites. We don't know who the culprits were. We have some ideas, but there were several culprits. The coprolites are fecal pellets left by an arthropod that gets preserved in the sediment. And these are loaded with spores. Remember, seed plants had not evolved, so none of these sediments have pollen. Seed plants were to come many tens of million years later. But some are lenticular, some are spiral shaped. And so we have very good evidence that these spores are being consumed. That doesn't mean that they were engaged in some transfer of pollen from one member of a plant to another member of the plant. But at least that feeding type is present. Now we kind of fast forward about 306 or 308 million years. So we're fast forwarding almost 100 million years to the Calhoun coal of Berryville, Illinois. And here we have, this is an enlargement of a section of this coprolite. And this is Florinites pollen. This is bisaccate pollen of a cordite, which is a kind of a sister group to conifers. It's an extinct group that went out at the end of the Permian. And here's the central part of the pollen grain. Here's a saccus, and there's a saccus. So it's a bisaccate, kind of like pine pollen. And this is another coprolite, like the one I showed you previously, totally loaded with Floronides pollen, which is the pollen of the cordite plant. You also have pollen. No, I'm sorry, excuse me. We have spores of tree fern spores, and they're found in the guts of these nymphs of these paleodictyopteroid insects, a large kind of dragonfly-like insect with piercing and sucking mouth parts. But this is the terrestrial nymph of it, and this is what the coprolites of this guy, or these something similar to it, look like. And this is made out of convoluta spora, which is a fern pollen. And we have other evidence from Europe in the case of the picture on the right. And this is a good example of a paleodictyopteroid insect that, although it's not the same species, but the adults look like this, and the nymphs look like that. Some of them get quite large. So now we go to the Permian of Russia, and this is really a very interesting development because in the late early Permian, we have deposits called the Chakarda deposit, and these are very small insects. These that I'm going to show you in the next few slides are about the size of a mosquito, but their heads have kind of a long proboscis. And I think that what's going on here is that they're probably inserting their these proboscises into the micropyle of the seeds. And I'll talk about that later, but gymnosperms produce seeds that have a kind of a tubular structure called a micropyle, and it secretes a thing called a pollen drop. And that pollen drop has very similar chemical components to nectar, to angiosperm nectar, including lipids, carbohydrates, and amino acids. So this is a lacewing, a very early, scorpion fly. It's in the Mecoptera. We also have one that's a lacewing, also mosquito-sized, with a very interesting proboscis. This is the compression fossil. It has these little stout spines at the end, reconstructed here. And it also is probably feeding on fluids from gymnosperms. Here's a third example, another scorpion fly. But this is really interesting. It forms its proboscis not by images of the maxillary segment, which forms a tube in the case of butterflies. But it forms it from the palps, which are subsegmented. And so those palps get conjoined and then they form the tube. And that's what we have right here. Now, this is an example of a beetle that does the same thing. It's a modern blister beetle. But here you can see the maxillary palps. And sometimes when these things get preserved, these palps separate, but they're conjoined in line. And here you can see another example of it. The plant that it was feeding on is a peltosperm, which is an early seed plant. It's extinct. It went extinct later in the Mesozoic. And this is what the fructifications of the female organ looks like in cross section. There's no reproductive structure that looks like that today. But it's interesting that these plants were being used in this way. Now, here's a completely different type of pollination. Also, all of these from the same site in the late early Permian of Russia. This is a mandibulate insect. So it has normal beetle-like mandibles, chewing insect. It's called a tiliard imbida, and it belongs to this order, the Nemediolestida. And it has an elongate abdomen, and it's loaded again with Floronides pollen. And this is what these cordites look like. They have long strap-shaped leaves, unlike conifers, their sister group. But the structures, kind of cone-like structures that contain this kind of pollen. And evidently, between the male and female organs. Let's look at some insect diversity patterns. So what got me started in all this? So this was a paper that I did with Jack Sapkowski when I was a graduate student at Chicago. And all we wanted to do was just plot the number of families of insects to geologic time and see what kind of pattern we get. So here's the vertebrate record at the family level. This was 1993, which kind of dates me a lot. We also looked at mouthpart structure. So part of my dissertation at Chicago was to look at the fundamental ways that mouthparts are structured today and to trace them back into the fossil record. Turns out that there were 34 fundamental mouthpart classes, and they show this pattern. This is these two curves are has to do with this is a better reading of the record, and this is a lesser reading of the record. And the interval is in the red here, it's in the red here. But what's noticeable is that during the ascendancy of the angiosperms, the record is flat, both for the mouthparts and for the insect diversity at the family level. And so we wondered, what would cause that? Why? I mean, the standard view would be that, you know, you had this explosion of angiosperms during the early Cretaceous, and you would think that the insect herbivores, pollinators, anything having to do with angiosperms, feeding on them, and the live tissues would have increased, but not, it didn't. So I also took it more recently, 2014. This paper was published that I worked on looking at insect diversity through time, but this time I segregated the families that are only associated with angiosperms, pollinators, and herbivores. And these are ones that are in the dark green are associated with gymnosperms, and in the light green are those associated with angiosperms. And it turns out that the plateaus are not that different. And this is the transition. And so this is what we're going to discuss next. This in the pink are ferns. So ferns don't really add much to this discussion. I guess at this point. In the pink. So let's go to the mid-Mesozoic. At the 4 pollination modes that we've documented. There are others, but I think 4 is enough for today. So this is called mouth cone. The mouth cone is a very unique mouthpart type that has the insect group that possesses it are called thrips, and they're in the order Thysanoptera. And it turns out that in the past, they were associated with gymnosperms as pollinators. So we'll look at this thing called polystrips. This is again from Oliva amber from Spain of late early Cretaceous age. There are amazing things. So this is what the fossil trips looks like. They're no more than a few millimeters long, no more than 2 or 3 millimeters long. So they're small. These are the things that you find in your flowers, that they look like little hyphens. They're moving around, especially at the base of the petals. And so we found this thrips, and it turns out that their wings have these very specialized seating. And it looks like as if they're threaded by a rope threaded with donuts on it. And so we call these ring seating. We've never seen this either in the modern or in the fossil record. This is the only place where we find this specialized seating. And around the seating, we find clumps of pollen. And here you can see clumps of pollen, and they're in the orange here. And but they're not found on the mouth parts or in the head or even on the legs. They're only found on the seating of these the wings of thrips. So what is happening here? Here's some more wing seeding with pollen clusters. It's like we think it's shuttling between the female and the male part of the plant, and that it's taking the pollen from the male organ, which is this thing, to that thing. Now, the host plant is an extinct species of nematophyte, distantly related to modern ginkgo, probably not in the same family. Called Nebesdiella. And as is in paleobotany, things have to become complicated. So we give names to each of these organs. So this is the short shoot. It has a name. The pollen has a name. The female organ has a name. The leaves have a name. The seeds have a name. So, but we'll keep it simple just to show this diagrammatically. So if you look at the history of thrips pollination through time, it looks like that there are three major seed plant groups that have been hit. So I mentioned the thrips ginkgo phyte pollination in the from Spanish amber, and that's gone. That's extinct. Both that type of thrips and its host plant became extinct at the end of the Cretaceous. Today, thrips occur in cycads in Australia on a thing called macrozamium. And then, of course, they're all over angiosperms. It's hard to avoid them. Here we have the history of pollination of one order of insects with a specialized mechanism for securing the pollen grains that no longer exists on insects. And it could be a kind of roughly equivalent to the branch hairs of bees. So this is supposed to rotate, but we weren't able to make the connection. But this shows you, if you can imagine this thing whirling around with these orange dots, and the orange dots are the clusters of pollen that occur only on the wings. You get maybe a few in the head. And so we were able, since this is an amber, we're able to three-dimensionally reconstruct the thrips insect. So we go to pollination mode 2. So we talked about the thrips and the punch and sucking type of mouth parts. These have mandibulate mouth parts, and the feeding style is not punch and sucking as before, but chewing. And we have these very interesting, also from Alba amber stains. It's a late early Cretaceous. You've seen this before. It's a false blister beetle. And this is kind of maybe a better image of it. You can see the beetle where it ended up. It impacted the amber here, and you can see the grains, and it's all cycad stuff. It's on the cycad. And this is a reconstruction. Now pollination mode 3, this is the big one. So this is the long proboscis pollination mode. The feeding style is through siphoning. And there are these 4 orders of insects in the mid-Mesozoic that participated in this type of pollination mode. There are additional ones today. For example, beetles have been, some beetles and some caddisflies have these kinds of mouth parts. There are these, I say 38, but there are these 43 species have been described that have this type of mouth parts. And this is a type of mouth part that originated at least 13 times. And we tried to figure out why that is, and I'll talk about that a mechanism by which that can be achieved. The host plants include things like conifers, bennettitalians, and also ginkgo phytes. So it's a wide host of all gymnosperm data, and it's mostly comes from the Middle Jurassic to the early Cretaceous, mostly Asia. Almost all of it comes from Asia. So let's talk about the scorpion flies, order Rhipoptera. So this is what a modern scorpion fly looks like. This is standard issue scorpion fly. This is a frontal view. So what's important is that this rostrum is actually part of the head capsule. It's rigid. And the mouth parts are at the end. These are mandibles. Here you can see one sticking out. And you can see the same thing on the side view. The business end is right here. And here's what an adult looks like. Relatively rare group. There are only about 500 modern species, which is almost close to extinction by insect standards. This is what a mid-Mesozoic long proboscis scorpion fly looks like. So the brittle part of the head capsule ends right here, and this is all part of the, in this case, I think it's the maxillary segment. No, it's the labial segment. It's the last segment of the head that is prolonged into a tube. And here you can see the feeding canal. And there's always kind of a structure at the tip that kind of interacts with the plant host. And here are the maxillary palps. They're just small structures at the base of this long feature. Here, this is an antenna, compound eyes on each side. And this is in the family Mesozoic. There's a second family called the Angiosperm that has different kinds of mouth parts. Well, it's the same long proboscis mouth part, but it's very structured differently into annuli, and a much larger tip with the mouth in this position. These proboscises are a little bit less than a centimeter long. The Mesozoics have somewhat longer proboscises by exceeding a centimeter and going as far as 2 centimeters. This is the third one. This is a mosquito-sized one, kind of like the ones in the Permian, lower Permian. And its proboscis is again very different than the other two. It's very smooth, and it's short. Here you can see it right here. And this is the pseudopolycentropotidae. And that's a tongue twister, but so there are 3 distinct families. And we think that there were 2 originations of the, actually 3 originations. There's another family that I haven't mentioned yet. This long proboscis morphology just within the Mecoptera alone, just within scorpion flies. Get a long proboscis morphology. So what some really enterprising evil people did is that they took the adult mouth, they took Tribolium, which is the flower beetle. It's one of these insect model evil standards, kind of like Drosophila is for flies, Tribolium is for beetles. And they did a knockdown test on these 2 genes, homothorax and extradentacle. And what happens is that the labial segment of the flower beetle actually loses its appendages. And the labial palps, in this case, can join into form a tube. It's not a long tube, but it's a tube nonetheless, and it's hollow. So in these 2, and this is a very relatively simple knockdown of using larval RNA, you can actually get a precursor to what a siphon would look like from a typical mandibulate insect. So, so we talked about scorpion flies. Now we'll talk about lacewings, order Neuroptera. These are amazing things. So they're convergent with butterflies. So this is a typical nymphalid butterfly. I think it's Bicyclus. No, it's not. It's Caligo. It's an nymphalid. They tend to be these really beautiful things like painted ladies, for example, that you see in the chaparral in California, for example. And they have beautifully developed wing eye spots, scales, very distinct kind of scales, and a proboscis. In this case, the alien is separated. That actually coils. And this is a thing from the early Cretaceous of northeastern China, thing called origramma. And it too has wing eye spots. And what's interesting is that they're in the same 2 veinal regions as this one. So that may be a very conserved evolutionary structure. And it has scales. It has all the other kind of butterfly-like structures, except for this one has a long ovipositor. Modern butterflies lack an ovipositor for laying eggs. So this is a good example of evolutionary convergence. But in terms of their ability to siphon fluids from a seed plant, they're probably doing the same exact thing, even though there's no overlap in geological time between these two lineages. So here's some examples of lacewings. They're quite large. So some lacewings have wingspans of 16 centimeters. So they're as large as the largest probably atlas moth. And so these are maxillary palps. These are very robustly constructed mouth parts, and they're probably going into really robustly constructed plant hosts. Maybe something like a Binatalian floral-like structure. So here we have another one. So this is the head region here that's enlarged here and drawn here, and it has a very long. Some of these proboscises got up to about 3 centimeters long. Now, this one from Kazakhstan, from Karatau, latest Jurassic, has actually pollen attached to its mouth parts. And of course, these things have scales, although they're differently constructed, but the scales that are analogous to those of butterflies and moths. So when you look at those mouth parts of that one from Karatau, these are the scales. They tend to be longer, but they have the same scale-like structure. And this is the pollen tetrad that is found on these scales. And we actually looked at geochemically the matrix versus what is shown on the scales and on the pollen, and they're very distinctly different. This is that belongs into the Chirolepidiaceae. There's no common name for it because it's extinct. And it features quite prominently as a host for many of the mid-Mesozoic pollinators. So let's talk about flies now, order Diptera. These are tanglevane flies. They're still around, mostly in the famed boss of South Africa. These tanglevane flies, the Nimistrinity, have long proboscises, but some are short and stout, and some are long and probably do very different functions. These are modern Nimistrinids. These are from the famed boss of South Africa. And these are deep funneled iris flowers, and the nectaries are down at the base at about this level. And these are gladiolus flowers, and again, the nectaries are probably at this level down here. And so this is an example of kind of one array specialization, if I use that word. There are moths like that, for example, pollinate Malagasy orchids that even have longer proboscises. But it's the same group that we find in the early Cretaceous of northeastern China. And now the moths, they have the poorest record of all, because moths don't preserve all that well. So sometimes we have to get the presence of certain lineages of moths from indirect evidence, such as leaf mines. So the leaf miner of this is a moth called a gracilariid, or leaf mining moth, that its adults are nectar feeders. Also, the structure of the hind end of the ovipositor is another clue to whether or not something was catasiphonate proboscis or not. So when we kind of put this all together, all these long proboscis insects, these are all these insects. I'm leaving out the moths, which would be small, a little bit smaller than this one, put in the same standard scale. So this would be 1 millimeter. Some are very robustly constructed and doing things that are quite different than, say, this thing was doing, this pseudopolycentropoid. This lacewing, and this is a fly, this is a mesocycid, and this is an anurettocycid. So they have different structures. Some are very hirsute, some are ridged, some are smooth, some are robustly constructed, others are more gracile. It's really an assemblage of insects that are pollinating some kind of gymnosperm with a tubular or funnel-like structure. This is kind of to put it in deep time context. This is the period of time that we're talking about. It's the Angiosperm radiation, or what I call also the Aptian-Albian transition. And these are the Permian ones that we found that I discussed earlier. And then there are also a lot, some of these are actually extant, they occur today, but a lot of these also became extinct. And a lot of them actually were once long proboscid, like some of the scorpion flies, but they lost that feature and long proboscid members. Pollination mode 4, this is probably the one that you may be more familiar with, because houseflies do this. Not that if you've ever been at a window and there's some sugary substance and you can see the vellum of a housefly palpating the surface, well, that's what this structure is. So there are these things called pseudotracheae that kind of lead into a canal that goes into the mouth. And these things are fluid feeders, and they do it by sponging. And but some of them also consume structures like pollen, such as this Drosophila on the right. This is a modern Drosophila, but also in the material that you just saw on the previous slide, we actually see our friend Plasopilus also in those sponging mouth sac devices. This kind of puts it all together, all these types of clades. We have 4 orders. I haven't included the Lepidoptera here, but they probably should be. Range a lot from Benetalians, conifers, it's Pyrolipidales, all extinct cycads and aliens. A lot of them come from Malabar amber, but they come from Daffodil, China, Karatau in Kazakhstan, and Alava in Spain. Material from Spain is amber, but all the other material is compression material. So what can we say about the plants? Well, remember I talked about the micropyle? So this is what a gymnosperm seed looks like, or ovule looks like. So unlike an angiosperm ovule or seed, they have these things called micropyles, and a nectar-like substance called a pollen drop is secreted. And the idea of it is that it kind of bubbles up to form a surface, and pollen transmitted through the air lands on that sticky surface, and then the bubble gets resorbed into the pollen chamber where fertilization occurs. Of course, the pollen develops a tube nucleus. And this is a standard, you find this in all gymnosperms today, cycads, Metalians, and in Ginkgo as well. But it's also occurred in gymnosperms of the past as well. So here's some examples. This is a Phaedra, commonly called Mormon tea in North America. And then this is this bizarre plant in Namibia called Welwitschia. There's only one species left of this Mesozoic lineage. So there's also a thing called Catonia, another seed plant group that's gone. It's probably survived into the Cretaceous. This is the pollen organ. It's called Catonianthus. This is the female. That's called Catonia. And if you took a cross section of it, what you have is these are the ovules, and this is the micropyle, and it secretes a pollen drop. Each one of these. Probably what happened is that the proboscis, long proboscis insect, secreted its proboscis here to access these pollen drops. Here's what they look like in cross section. These are the seeds. This is the associated foliage. So there's this lip at where the insects would secrete its proboscis to access the pollen drops secreted by the ovules. Now, this thing is very complicated. I'm going to have to urge you to be patient with me. This is the most complicated pollination thing I've ever seen in any angiosperm, and probably in any seed plant at all. This is a chirolipidiosis cone called albinia. And each of these is a reproductive unit. It's kind of like a scale of a pine tree, except much more fleshy. And these things are funnels. And this is what it looks like in cross section. So this is the axis of the cone-like structure. This is the ovule with this little flap sticking out, and this is a funnel. And but the micropyle is here, and this is where the pollen drop would be. But there's a connection between the funnel and this area called the pipe. That's the formal term, the pipe. Now, this is Catonia. We talked about Catonia. There's also a thing called Lepidostropus, which is a kind of a relative, Cheslowski alien. And these ovules are canted backwards, and the pollen drops are here, but it's a bivalve-like structure in which there's a channel through which a long proboscis insect can insert its proboscis. And then this is another group of extinct gymnosperms, a pentoxylalian, and this is a micropyle, which actually can be quite substantial in terms of its width. So this is just an example of the tubes, channels, micropiles, and salpinx stars that you see in this whole array of gymnosperms during the mid-Mesozoic. So you have these other things called Bennettitalians. This is a female part. It's called Williamsonia. And some of the small ones just kind of occur with a kind of oppressed bracts and with the kind of a mound of ovules each with secretory structures. And somehow insects will get into this. At least that's the hypothesis, and access the secretory structure. This is the male organ with these pollen sacs with pollinia within the sacs, arranged kind of like in pairs along the inner surfaces of these outer bracts. And this is another example of the Williamsonia. These things also have a lot of damage associated with them, insect damage. So let's look at the mid-Mesozoic plant, and this is the pollination from hell. So here we have that thing that I showed you before, the albinia. So that's the female organ, and this is the male organ. It's a smaller cone-like structure with these pollen sacs. So if you were to take one of these structures and section it in half, this is the funnel. And the funnel, especially at its base, is loaded with these nectary-like structures. And this is a photograph of a nectary-like structure. And around its orifice, towards the outside, you have these trichomes. They're very fat and kind of fleshy. And so this is probably an attractant, as are these nectaries, which could be a food source for one of two kinds of insects. Either a long proboscis insect sticking its proboscis in here, or a very tiny mosquito-sized insect moving all the way in. So once the pollen is deposited here, either the nucleus would move, the tube nucleus would move through the pipe and into the micropyle, which is an amazing journey. I find that just kind of incredible. But we do know that the pollen of these guys were being consumed by grasshopper-like insects and also by these flies that I showed you previously. So that's kind of a plant end of things. For the insect end of things, this is another scorpion fly group called the Dualidae, although that name may have been preempted. This is what it looks like. It's amber. This is an image of it, and this is a pump. And you have both a cerebral pump and pharyngeal pump. And the pharyngeal pump is kind of like a piston that would secrete salivary fluids out, and the subaerial pump would have the opposite effect of feeding fluids into the esophagus eventually. So it's pumps working in opposition. And you can see this detail in Spanish amber, and it's amazing. We had to, my student Shalon Dean worked this out. It's quite a structure, and you can see some of the gory details. This is the valve of that hypopharyngeal pump, the one that secretes fluid out, and the other pump is up here in that position, bringing nutrient-bearing fluids inward. And this is an example of the hypopharynx, which is another mouthpart region. This thing was feeding on fluids. Well, in amber, you can actually see the ends of these siphonate mouthparts with fluids coming at the end. So here's fluids. See the salivary channel. So here's a reconstruction. So here's the hypopharynx that is exuding fluids outward, and coming in is the food tube bringing fluids of nutrients into the insect. So the transition to angiosperms rests on this really interesting find, also from Alva amber. So these three resin pieces involve cycad, pinco-phyte, chirolipidiaceous amber, pollen in these amber associated with these short-winged flower beetles of the Chitorididae. But in one piece, we found water lily pollen, and it's in this and in this. And these little dots indicate where pollen has been identified. And that thing is first, it now has the name of Clade Nymphopollinitis cinnamoniensis. And that's the first example, the earliest example, I should say, in the fossil record where we have evidence for this transition between Gymnosperm pollen and the first evidence for Angiosperm pollen being used in a pollinating insect. What's amazing is that it took so long. And so these Gymnosperm associations were kind of well established. And so now, but what's also interesting is that the species of short-winged fire beetle that is responsible for the water lily pollen is in the same genus as the ones that are responsible for the Gymnosperm. It's really a shift within a genus from Gymnosperm pollination to Angiosperm pollination. And here we can see the Gymnosperm pollen. This is typical Cicadopides. And here we can see the water lily pollen and Angiosperm here. And it's this guy. There are two different species of this genus of short-winged flower beetle. So this is just kind of illustrating the changes in modes. This is our Calligrammatid, probably on a Venetitalian. And then this is the earliest appearance of an Angiosperm pollinating insect at this level in the upper part of the lower Cretaceous. Even though Angiosperms have been around probably for about 30 million years prior to that. This kind of shows you the diversity of insects, the diversity of host plants, the diversity of the pollen, and where they came from. And these data are increasing almost every year. Every year there's something new coming out of this. Here's another way of showing the data. So these guys are extinct. And see, these are the Angiosperm pollinating insects, and then these are the Gymnosperm pollinating insects. There's two insect groups. I mean, there are many insect groups, but I'm showing here two that are associated solely with angiosperms. One are the bees. This is probably a late Cretaceous one from New Jersey, amber. Also the butterflies, and this is from the Green River Formation. I think, no, it's from the Forest Sand Formation. And they typically are much later in occurrence in the fossil record. So I'm going to kind of wind this up with evolutionary cohorts. This is another example of the diversity of pollination. These are all insect groups that pollinate plants. There are others than the four modes that I mentioned. You've seen this before. Just to reemphasize, there's this Gymnosperm kind of domain of pollinating insects and herbivorous insects. This also includes herbivores, and then an Angiosperm domain. So what's going on? I think there's four things going on here. So we have an ecological evolutionary cohort in which Gymnosperm associations became extinct at the radiation of angiosperms. There's a second ecological evolutionary cohort in which there was a huge bottleneck and there are a few survivors. And there are examples of these. This is the Gymnosperm, for example. The thrips that I mentioned earlier. These are and then there are those that made the transition, such as these beetles that I mentioned previously. And then there are ones that never had a history of Gymnosperm associations, such as bees. Now, this is the last slide. Also have evidence from the ichnological record. So these are flowers. I think there are several flower morphotypes represented in the Dakota Formation, and these are petals, and they have all kinds of damage. We have names for these kinds of damage. Here's a notch taken out of the petal. Here are some punctures. This may be a gall, and there are other structures, usually around the edges of these petals. So we know that these flowers were being consumed by what we call herbivores, that are insects that are feeding on petal tissues, which is exactly what happens today with many pollinators. This is indirect evidence, but it's very, very, very supportive that at least by the Dakota Formation times, which is at the end of the early Cretaceous, almost at the very end of the early Cretaceous, that there were already angiosperm associations. So that, I'll end with that. Thank you.
Thank you so much for such an interesting talk, Conrad. And we are open for questions.
That was wonderful. I really enjoyed that. Yeah, I know nothing about plants, but the fascinating thing about Angiosperm pollinators. What do you like labile? Did it take that long for the Angiosperm for the pollinators?
That's a very good question, because I wondered about that by myself, and I think there could be several solutions to that. One is that intimate mutualisms acquired by faithful pollinators takes time to develop. And with the Gymnosperms, they had all the way back to the Permian to develop these, if that's what they are, co-associations, or maybe even co-evolutionary mutualisms. And it may be that it takes this long to build up these kinds of co-evolutionary mutualisms in angiosperms. They really didn't become ecologically dominant until about years before our. And so I think that that's one possibility. Maybe part of that is also that also build kind of a community of mutualists. For example, ants protect plants from predators because the ants give, the plant host gives ants something in return. And maybe those took time to develop. That ants are very seminal in modern pollination nutrients. It's not because of their pollinators, but because they defend plants. And so maybe that's another issue that needs to be addressed. And it could be that, and then, you know, our friend of Albania, the Paleozoic, is that they just didn't have a better system than Angiosperms. So while they went extinct, and almost all of them went extinct by the end of the Mesozoic, it took time for them to for Angiosperms to replace.
Well, any other?
Pattern. Do you see any sort of pattern in terms of islands or? Yeah,
I think so. Wasn't that the same fire?
I think that it has to do with where all these major deposits are. So almost all of them are Eurasian. There's only one example of a long proboscis insect from South America, for example. There may be some evidence for. It's just a rumor at this point, but it's possible. But other than that, it's a Eurasian phenomenon, and it could be that during that time was in a favorable latitudinal. It's not preserved in North America. I mean, Mesozoic insect deposits in North America are horrible. There's nothing known from the Morrison Formation virtually. And even in the Cretaceous even though we know that they're doing all kinds of damage, as we can see on the flowers, but they're not getting preserved. So there's this differential preservation also between plants and insects. So it's kind of a multifaceted problem.
Those thrips with the CK on their wings that collect pollen, what are the advantages for the thrips to collect that pollen?
So in the ovulate structure, I didn't. So what's interesting is that our image of these ginkgophytes, modern ginkgo, it's wind pollinated. All these Mesozoic ginkgophytes are insect pollen. So our image of a typical modern ginkgo biloba is really an outlier. And it probably survived because of its wind pollen. So these ones in the Mesozoic have things like nectaries and other structures that attract the insects. So that's probably what the thrips are getting out of it. Also, the other thing is that it also formed probably an area for breeding. So it's a safe harbor in some ways.
I'm just wondering about the size of pollen. Usually when you have insect pollinators.
Yeah.
And whereas wind is smaller, you see that.
Well,
that they're-
yeah. So the Crassopoulos pollen from the Chirolepidiaceae, the individual grains of pollen, but they also occur in tetrads. So the functional unit is probably the tetrad in that particular plant family. There's other indication that some of these other pollens have maybe substances on the surface that may be that may allow transfer. And modern cycad pollen is consumed. And so and modern cycad pollen is virtually indistinguishable from Mesozoic cycad.
Any question? Okay, so a big round of applause.
Thank you.
Thank you. Thank you. Because we may have another